This session is going to start in 20 seconds. At first we give intro, later on it starts. Audible, sir?
Yes, audible. But some music is coming. Hello. Some background music is.
Good morning, everyone, and welcome back to day 2 of our virtual FDP. Yesterday, we had some great discussions on implementing the quality education in pharmacy by integrating technology and pharmacovigilance and adverse drug reaction management. And today, we are building on that momentum. Today's sessions will be focused on role of regulatory affairs in drug development and bioanalytical method validation and recent developments in pharmacokinetics and pharmacodynamics in the aspects of plant-derived natural products. And we are fortunate to have experts like Dr. N.Kannappan and Dr. Tulibali Shyam to guide us through these important discussions. Now, let me invite Dr. Ennappan Garu into the studio, and I'm going to give a brief intro on Dr. N. Kannappan Garu. Dr. N. Kannappan Garu is a professor in the Department of Pharmacy at Annamalai University and bringing 28 years of teaching experience with a specialization in pharmaceutical analysis. He qualified for GATE in 1994 and served as a JRF during his postgraduate studies. Dr. Kannappan has authored 98 research papers in both national and international journals and has presented at 33 national and international conferences and symposiums and workshops. Under his guidance, 38 MPharm students and 17 PhD scholars have completed their degrees, and 6 of the scholars were pursuing under his guidance. He has successfully completed one major, one minor, and 3 major research projects worth 54 lakhs funded by various agencies. Additionally, he has delivered over 20 special lectures at various pharmacy colleges. Dr. N. Kannappan serves as a member of Board of Studies for multiple universities and reviewer for several prestigious journals, including Journal of Chromatographic Sciences. He is the author of the book entitled Handbook of UPLC, which is published by Lulu Press, North Carolina in 2023, and authored a textbook of pharmaceutical analysis, which is published by Shashwat Publications, India. Now, let me invite Dr. Kannappan Garu to share his insights with us. We welcome you, sir.
Thank you. Thank you, Divya Yada, for the given opportunity. Thank you, management of Institute, for the given opportunity. Can you hear me?
Dr.
Yes,
Kannappan.
Okay. But I am getting some feedback, means I'm getting some echo.
Oh.
Whatever I am talking, I am getting.
Sir, it is audible, sir. Before your session, my principal talks a few, delivers a few.
Your words are audible. But whatever I am talking, I am getting back echo.
No, sir, it is clear here, sir.
Okay. Then I will start. Okay.
Sir, before your session, no, sir, my principal sir gives some few words, sir.
Okay. Welcome.
Sir, Dr. Kannappan, sir. Welcome, sir. Welcome to the session. This is about Friday session, sir. This is second day. I'm very, very happy to have such a senior person like you to our session.
Thank you, sir.
FDP. This is given as a very good platform for all of us to meet on this online. So I request you.
Sure, sir.
Please, sir. Thank you, sir. Please.
Sure.
Please.
Thank you, Principal, sir. I am starting. So, role of regulator. The topic is role of regulatory affairs in drug development and bioanalytical methodology. Nowadays, regulatory affairs plays a major role in drug development. In those days, we used to say the drugs R&D department and other departments are involved in the drug development. The drug regulatory affairs plays a major role because regulatory affairs is also a recent importance, and people are not aware about this area. That's why I wish to give you the information. The slides are moving. Okay. I hope that the slides are moving. Second slide. The regulatory affairs plays a vital role in the journey of product from the laboratory to the pharmacy shoulder. So it is a starting from discovery to the market. And because the area will be from pharmaceutical or biotechnology companies expected huge amount because it spent nearly 5,000 crores for bringing a simple molecule into the market, and also it requires minimum 12 to 15 years because it enters into a different process, and it enters into a different area. And the new pharmaceutical product has to undergo the clinical trials and several important regulatory approvals. So without regulatory approvals, it is not available in the market. So no new product enters the market without any new drug approved from the respective competent regulatory authorities. So in USA, US FDA is as well available, and in UK, Medicine and Healthcare Regulatory Affairs is in UK, MHRA, and in Australia, EPA, so Therapeutic Goods Administration, and in India, as we know very well, Central Drug Standard Control Organization (CDSCO), and in Europe, European Medical Agencies. So these are the major regulatory authorities in the world, and so they have each and every country the regulatory affairs, regulatory regulations have different regulations. Regulatory authorities have a different regulations. So once if you fulfill these regulations, we can market our product in the respective countries. And the process involved in this drug discovery will be starting from the identification of the candidates, and it may be a traditional medicine from the natural origin, or it may be from synthesis. By the knowledge of the traditional origin, we can also synthesize the chemical compound or in silico drug design. Nowadays, a major role of the in silico drug design by computer drug design is available. After the isolation of the chemical entity required for the pharmacological activity, then we have to characterize the structure of the compounds by using the IR, NMR, and mass spectra, and then pharmacological screening and assay for therapeutic efficacy, bioassay for therapeutic efficacy. Then preclinical testing is important in the animal models, and then clinical studies including regulatory approvals for the commercialization of the new chemical entity and new biological entity. So these are all the different processes involved in the drug discovery. So in this area, nowadays we include the regulatory affairs also. So drug discovery starts from preclinical testing, clinical trials, phase 1, phase 2, phase 3, and product sale. And then preclinical and clinical trials starts from after synthesis of the new chemical entity from innovator, and the preclinical studies files IND. So the before going to the preclinical studies, we have to file the investigation new drug application to FDA for conducting the clinical trial. The FDA reviews the data of IND and grants permission to conduct the clinical trial. So they will verify this data which is given in the bracket. The preclinical data, chemistry means chemical nature of the compound, manufacturing details means what is the formulation, dosage form, investigation protocol, how we are going to investigate, what is the investigation protocol, details of the investigator, concern of the participants and other agreements. So these are all the different areas to be covered. And phase 1, phase 2, phase 3, and phase 4 is the different clinical trial phases. And preclinical evaluation also demonstrating pharmacological effect of the lead molecule using the suitable animal model. And the disease oriented also organ oriented drugs are employed to provide the information about the preclinical data. And it includes toxicological studies, pharmacodynamic studies, pharmacokinetic studies. So for establishing these three data, we are performing preclinical data. So now we'll come to the different stages. It requires minimum drug discovery of the preclinical trial will take minimum 3 to 4 years, and then the x-axis is phases, y-axis is years. And phase 1 clinical trial will have another 2 years, and take another 2 years. And phase 2 clinical trials will have another 2 or 3 years. And then finally, phase 3 clinical trial. Once the phase 3 clinical trial is over, then we can market the product in the, we can distribute the product in the market. and the FDA review and approval. After that, we can go for the phase 4. Phase 4 is post-marketing surveillance after the marketing into the restricted area. And phase 1 phase 2, we see in detail about phase 1 phase 2 and phase 3. And phase 1 will be a healthy human volunteer with a small group of 20 to 100 volunteers, 6 to 9 months, around 1 year, to establish a determination of the pharmacokinetic absorption, distribution, metabolism, and excretion, and difference between animals and humans. Because we have a data, preclinical studies before starting the phase 1. So we have to compare the preclinical data with the phase 1 data. And phase 2 will be diseased population, natural or induced. 20 to 200 volunteers will be involved, and 6 months to 3 years. And in this set, determine the effectiveness and the safety of the drug in patients with the target diseases. Because the phase 2 will be mostly the disease. For example, if you want to establish the drug for anti-diabetic, then we have to catch the diabetic patients as a disease population. The phase 1 is not a disease population. Phase 1 is a healthy human volunteer to find out the pharmacokinetic difference between the animal and the human. And phase 2 will be the diseased population. So there are two phases are there, the early phase and later phase. The early phase will be 22 to 400 volunteers, single blind study, and later phase will be 30 to 300 volunteers, double blind study. So single blind study is the single blind study is there are difference between the single blind study and the double blind study. So single blind study is the study will be conducted by the innovator, and the study will be conducted on the subject. So the subject may not know what purpose the study is going on, so that we can avoid the bias. And double blind study means even the operator or the physician or the chemist who is giving the drug, and the patient who is receiving the patient or the disease population who is receiving the drug also will not know that thing. So this double blind study is mostly it is used to avoid the bias. So that's why the single blind study, double blind studies are used. And phase 3 will be 200 to 1,000 plus volunteers, 1 to 4 years, to establish the safety and efficacy of the drug candidates. And these are the phase 1. Phase 1 to establish the pharmacokinetic profile, determination of safe and tolerable doses, and preclinical toxicity. And then phase 2 will be safety and effectiveness of the test compound, and recording of short-term side effects in healthy human volunteer, health impaired patients. Actually, phase 1 is used to establish the predictable toxicity and also pharmacokinetic ADME. And phase 2 will be to find out the safety and effectiveness of the compound, and then determination of the dose range. You can fix also the dose. What is the dose for the ailment, for the severity, according to the severity of the disease. And phase 3 will be recording the label information, and recording of the risk benefit ratio, and also recording of the less common side effects will be measured. And phase 3 will be several correspondence between the actually when we are filing, if you want to file the NDA, new drug application, IND, investigation new drug, we have to find out we have to apply the application will be submitted to the FDA or any other regulatory agencies. So they will be verifying the data. So for that, there will be the several correspondence will be between sponsor and FDA for clarification. So once phase 3 is completed, sponsors files NDA. So new drug application, they can apply for the new drug application to FDA for marketing the drug in the respective country. So if FDA is satisfied with all the details, so without certification, it will not give the details. So if FDA is satisfied with all details, then allowed to enter into the market with the new drug status. So that's called controlled marketing. So that is called your phase 4. It will enable to give information about the phase 4 studies. Means in the phase 3B starts before approval of the additional safety data. So we can find out the cost and effectiveness of the drugs by the phase 3B. And once the controlled marketing is done for the phase 4 studies. And phase 4 is post-marketing surveillance. That is called restricted marketing. And it's a standard one it's started once the drug is marketed. So the post-marketing surveillance is called post-marketing surveillance. So it is a restricted marketing. So around 5,000 to 10,000 people. So we have to catch some disease-oriented physician, and we have to give the drug to their patients of the target disease. So if there is no specific time period, so we have to find in this we have to find out the less means very rare side effect. So sometime we may get the very rare side effects from the any one part of the races or any part of the country, so that we can document that data also. That is very important data for the filing of regulatory consideration. And manufacturers should gather the report such new information to the respective regulatory authorities until it is released to the unrestricted market. So the unrestricted market means it's a huge marketing from the top to bottom of the country. But restricted market is only a restricted area. So if anything went wrong, you can get back the drug immediately from the restricted market. And these are all the different areas of drug discovery. It starts from the molecular biology, and then biochemistry, medicinal chemistry, biopharmacology, biopharmaceutics, and nowadays it also includes the drug regulatory division. So structural biology and molecular modeling, and then drug regulatory division. And what are the functions of the regulatory affairs department will be to keep tracking of the ever-changing legislations, amendments, and existing rules and regulations. Because every day rules and regulations are changing, so that we should be aware about the changing regulations and amendments. And also we need to study the people, those who are entering into the regulatory affairs department, they must know these points. So we need to study the process of drug development, and they should know the data generated during the preclinical and clinical data. They should know the QC procedure of the particular drug. They should know the safety and efficacy of the product. So these are all the awareness about the regulatory affairs personnel. And regulatory affairs personnel give statistic and technical advice to all the concerned department, because R&D, manufacturing, marketing, relations, all the departments they'll be in connection. So role does not stop after the getting approval. So majority of the regulatory submissions requires maintenance of the existing marketing practice, and modification, the amendments, and updating product literatures, etc. So what is the role of regulatory authorities? So they should know about the communicative competence, and also both oral and also analytical abilities and presentation skill. So these are the basic requirement of the people, those who want to become a regulatory affairs person. Because nowadays there is a PG program is also available in M.Pharm regulatory affairs. So you can also get the information from the by studying that course. So there is a separate course is available, and even pharmaceutical quotation is also most related to the regulatory affairs. And these are some of the important area of regulatory affairs of USFDA. So I want to touch up little information about these all four areas. First one is a drug master file. So that's called your means which is a file which is submitted to the USFDA for keeping the information. And if anyone wants to buy the product from our product, so that will be disclosed to the particular person who are buying the product. And the investigation new drug application is a first application for the investigation of the new drug. And the new drug application is the application for the marketing of the drug into the country. And the abbreviated new drug application will be once the patent period is completed, then we have to the thing will be open for all, and anyone can manufacture. That is the abbreviated new drug application. So anyone can also manufacture. Patent period will be over, so that that the innovator will be disclose the information about the product. And one more thing, the generic product will be emerged after the ANDA. And drug master file. So the drug master file is a submission to the FDA, and it may be used to provide confidential data. It's actually the confidential document, and it will be either a facilities, process, or articles used in manufacturing, processing, packaging, anything you can make a drug master file. Either we can drug master file the facility, can drug master file the process. They can make a drug master file of article, so that anything can be edited. The information contained in the drug master file may be used to support an investigation in drug application, and it used to support IND and also ANDA, NDA and also ANDA. So actually, the drug master file is nothing but the detailing of the product, detailing of the facility, detailing of the process, detailing of the article, detailing of the packaging material, everything, so that the third party, those who wants to buy, they can verify the quality of the product from the USFDA. So that is called your master file. The information contained will be useful for the IND, NDA, and ANDA. And there are different types of DMF. So the type 1 is manufacturing site facilities operating procedures. Type 2 will be drug substance intermediate materials used in preparation and drug product. And type 3 will be the packaging material. Type 4 will be excipient, colorant or flavor or essence or will be. And then the type 5 will be the except for reference information. Because we can get also information from the NDA and we can make one more file as our own DMF. An investigation program. So IND will be once if you want to investigate any new drug, you have to file the IND. So the terms are investigation drug or the investigation drug and the investigation medicinal product are considered to be the same thing. And this is there are different types of IND. Investigator IND, so it will be the submit for the new indication for the old drug. So if I give you the example, you are able to understand. So the aspirin previously they were using for only for analgesics in those days, 90s and 80s. But after around 2000 or around after 95, 1995, it is used for the uh, anti-epilepsy disease because it is used for the reducing the blood thinning. It is used to blood thinning agent. So there is a new indication for the old drug. If you want to go for the investigator IND, any new indication for the old drug also will be the considered as new IND. An emergency IND. So emergency IND, I think we have an emergency situation. So we had emergency patients in around 4 years back. So that is Corona. During Corona, this emergency IND, if you want to investigate any new drug which is useful for any epidemic or endemic in the world, so that we can go for any emergency input. So it will allow the shorter period for the verification and they will give you they will ratify the information after the product is invented. So that's called emergency IND. And then treatment IND is submitted for the experimental drugs by using the particular disease, a particular property, a particular disease, mainly for the particular disease by the physician of the particular their own patients. So this is a flow diagram of the investigation IND submission. So you can able to see the different parts, medical, chemistry. So that we have to file the case, we will file the information, and the information will be verified by the CDER. So reviewed by the Central Drug and Evaluation Research. So Central Drug and Evaluation Research laboratory will be there in the US. So they will verify the information and they will give the information. They will give the approval for the IND. So they will verify the medical data, they will verify the chemistry data, they will verify the pharmacology data, toxicology data, they will be verifying statistical data. So we have to give the sample also, so that they will be verifying the chemical test, physical test, biological test, microbiological test. So if there is any problem, they will give back. The loop will be there. So again, we have to rectify the problem. Again, we have to submit. So for the review, they take some around 30 days to 90 days for the review. After the review, they will be giving either feedback for any correction, or if the review is okay, then it will be processed through the data. So next, a new drug application. Next the process will be NDA. So once the IND is completed, then we can file for the NDA for marketing. It's a critical component in the drug approval process. The US FDA require the drug sponsors to submit an IND and NDA review before the new pharmaceutical can be approved. So if anything you want to market it, you have to get the NDA. And this is a program for the NDA. They will verify the medical, pharmacology, chemistry, biopharmaceutical data, and then even statistical data, microbiological data, everything will be verified. If anything they want to verify, they want to check, or they have any inquiry, then it will be given back to the innovator. Then innovator will will resubmit the data after the correction or after the revision. So this is the NDA action. The next approved application, then ANDA. So the ANDA is used for the patent. Actually, patent period is around 15 to 20 years. So once 20 years patent period is expired for the product that has been on the US market or even in any other country also, and the company wishes to market a copy. So if any other company wants to market a copy, then we can make it. So in the US, the drug patent lasts 20 years. So after that time, the manufacturer is able to submit any other manufacturer, means not a same manufacturer, any other manufacturer is able to submit an abbreviated application for the product. So the recently around 2019, anti-diabetic drug patent was over. Immediately the next day, more than 8 to 9 companies have entered into the ANDA market. That's called generic market. So ANDA is nothing but generic, but other manufacturers, if they give any brand name, then it will be called as a branded. So the advantage of the ANDA will be, or generic market will be, the cost of the product will be reduced. The cost of the product will be reduced. And sponsors of the ANDAs request to provide the information. So what are the information required from the ANDAs to do? Approved drug product with the therapeutic equivalency evaluation. So it should be equal to therapeutic evaluation, and also it will be the bioequivalent, therapeutic equivalent. Everything should be equivalent, and also dose equivalent also. So the same thing will be duplicated by the other manufacturer. So if you want to get the information, you can get the information from the orange book. That is approved product will be available in the orange. The orange book rates based on their therapeutic equivalence. For a product to be considered therapeutically equivalent, and also the pharmaceutically equivalent, the same dose, same strength, and same dosage. Bioequivalent will be the rate and extent of the absorption. So significantly different from the rate and extent of the absorption of the drug, which is to be interchanged. So the it should be bioequivalent now. And this is a flow diagram of the ANDA. The same thing will be followed further in IND, NDA, ANDA. So there will be CBR or OGD will be verifying the central drug evaluation laboratory will be verifying the data which we submit. Once all the data is verified, once all the data is perfect, then they will be proving the data. And in India, as you know very well, CDSO is controlling the regulatory department, and the central government will have a CDSO, Central Drugs Control Organization. So the DCGA, the Drugs Control General of India, is the director of the head of the institute, and then there will be a headquarters Delhi, and the zonal offices are there, including Chennai, Mumbai, Delhi, and Bombay, and also other places, Bangalore and Hyderabad. And in sub-zonal offices are there, the Hyderabad, Ahmedabad, and Goa, the some places are there. So port and airport offices are used to restrict the import and export of the drugs. So the many ports and airports are there. Chennai airport and ports, then your Calcutta airport and port, then Mumbai airport and ports, like Cochin airport and ports, Visakhapatnam port. So all those things are there for monitoring the import and export of the drugs. And laboratories, many laboratories are used to find out the quality of the drugs by using the testing. The 6 CDL, Central Drug Laboratory, and then CDTL, Central Drug Testing Laboratories, RDTL, Regional Drug Testing. Each and every state will have RDTL, Regional Drug Testing Laboratories, and IPC. These are all the organizational chart of the CDSO. The CDSO controlling these areas. There will be IPC, Indian Pharmaceutical Commission. They will form the IPC. They will be controlling all the laboratories. So Central Drug Laboratories, CDK, Central Drug Testing Laboratories, and all those things. And then even it is also implementing the Schedule M1, M2, and M3, Schedule D, U1. So these are all the related to manufacturing of the drugs. In this, I want to emphasize only the thing will be M3. All the drugs, all the other are already already there. But in this, one more thing is a draft new bill has also been given in the thing. The title is the thing is that so draft of new bill in 2022 they made it, but still it is not approved by the Parliament of India. So it is waiting for the approved by the Parliament of India. So that's why it is called bill. Still, it is not become an act. It is called a bill. 2022 new drugs, medical devices, because it is going to become Drugs and Cosmetics Act. Previously, we used to call it a Drugs and Cosmetics Act 1940 and 45. Now it is going to become a Drugs, Devices and Cosmetics Act 2022 or 24. I think in which year it is going to be implemented, it will be that year will be given. So these I want to emphasize only the thing will be Schedule M3, medical devices. In the medical devices, before 2017, medical devices has not comes under the Drugs and Cosmetics Act. So since the medical devices does not come under the Drugs and Cosmetics Act before 2000, before 2017, the costs are very high. And in 2017, 18 January onwards, medical devices are entering the Drugs and Cosmetics Act. So once the Drugs and Cosmetics Act, once it is entered into the Drugs and Cosmetics Act, the cost of the medical devices. So medical devices come from the manometer and even thermometer. Everything is comes under the medical devices, starting from the small devices to the major devices like the heart stents. So previously the heart stents were the 2 lakhs, 3 lakhs rupees costed. Now it is becomes reduced to 20,000. So this is one of the major breakthrough in the medical and Drugs and Revision of Drugs and Cosmetics Act. And then major responsibility is to conduct training programs for the regulatory officials, approve licensing of the manufacturing devices, and regulate the clinical regulations in India, to regulate the standards of imported drugs and other publication of the Indian. And these are all the other functions, regulate control over the import of drugs, approval of new drugs, and then meeting of drugs consultative committee and the technical advisory board, and licensing of the approval, licensing for approving blood banks, large volume parameters, vaccines, regulatory DNA, RDNA, and products and some medical devices. So the amendment of the Drugs and Cosmetics Act of the rules, because nowadays they are going to amend the Drugs and Cosmetics Act. So banning of drugs and cosmetics, and if any drugs which is found unsuccessful, then it will be banned. And the grant of testing licenses, personal licenses, and also NOCs for the export and testing of new drugs. And the state government licensing authorities will be licensing of new drug testing laboratories, approval of drug formulations for manufacturing, monitoring of quality of drugs and cosmetics, and then recall of substandard products. So if any drugs, because recently many drugs are, I think you might be seeing them news and newspapers. Many drugs are recalled into substandard, into the not of quality, not of standard quality, NSQ. So it is called the NSQ, not of standard quality. So due to the not of standard quality, I think recently some around 15 days to 1 month back, some around 43 or 45 drugs are recalled, and they given a notice, and then they are planning to recall. The investigation and prosecution in respect to the legal process, and pre and post licensing inspections, and these are all the state government licensing authorities or state government regulatory authorities. Duties of the state government regulatory authorities. And this is a circular which is notice given in 2022 July for the amendment of the new Drugs and Cosmetics Act. Before amending, there is no time to give suggestions from the publics. So they given a time from 10 days to 25 days, given a time, and they got all the reply from the people. And the regulations, the major provisions of the new drug bill will be it seeks the regulate the e-pharmacies, because nowadays e-pharmacies is a new trend. If you order the through the app, you can get the app. So there is a threat for this pharmacist, for this e-pharmacies. The e-pharmacies, there is a threat for the jobs of the pharmacist. So they are going to regulate the e-pharmacies and medical devices, and they are going to regulate the medical devices in the strict manner. And there are a lot of medical devices lists are there, type 1, type 1, type 2, type 3, and different ABC. So many devices, so many different types of devices are there, and provides for the penalties and imprisonment for failing to pay the compensation to injuries to the death. Because we have seen the clinical trial in the beginning slides. So in the clinical trial, many volunteers are involved. So if any the volunteers which is involved in the clinical trial, who is involved in clinical trial, if they went for any, if they got any injuries due to the clinical trial, there was no there was only insurance, but there was no any compensation in the Drugs and Cosmetics Act. And now they are going to bring into the Drugs and Cosmetics Act to implement the this compensation for the participants. Means clinical trial. So the next Drugs and Cosmetics Act 1940, for the first time regulations were conducted because it's around 75 to 80 years old. So that it is going to be implemented. It's going to revise it. It is going to be revised in the short while. But the bill was drafted in 2022, which seeks to replace the previous Act 1940 and 40. And in this, what is the new bill, Cosmetics Bill 2023, where Drugs, Medical Devices and Cosmetics Bill 2023, they are bringing Ayush, that's called already Ayush is there. So in that, they are going to be Sovaric. Sovaric is nothing but from Tibetan tribal medicine, medical devices, or medical thing, like traditional tribal medicines. They are also going to be the Sovaric and homeopathy in the Drugs and Cosmetics Act. So new definitions also. Draft bill introduces various new definitions or provisions like bioequivalence, biological space, clinical trial, clinical space, and all those things. Okay. The next penalties are going to be revised. Compensation for the people those who are participating in the clinical trial will be implemented. And also e-pharmacies, as I mentioned earlier, the permissions has to be taken to operate the e-pharmacy, and there should be a pharmacist to monitor the prescription. So everything is going to implement it. So no person shall himself or any other person by behalf of selling, start or exhibit or offer the sale or distribute any other drugs to the online pharmacy without the pharmacist. So we are going to give a license for the pharmacies, e-pharmacies. And medical devices testing centers also going to improve because if the medical devices come under the Drugs and Cosmetics Act, it becomes a rules and regulation. So that the provisions have been interpreted to designate or establish the medical devices testing centers. So that actually the recently our finance minister has given in the entering union budget before the election. So it provides necessary fund for establishing the area of medical devices testing centers, and they propose to include a new PG courses for medical devices. So there, I think in future there may be an M.Pharm medical devices available, M.Pharm branches may be available, so that we can also compete with the people those who are coming from the engineering background. So because in engineering nowadays they are planning to recruit the engineering graduates for the testing the medical devices. So if we have a inform program with the medical devices, then we can enter into the medical devices testing centers. Next thing will be the bioanalytical method validation. So this is an additional part of this one. So actually the bioanalytical method validation is different from the normal method validation as per the Q2A and Q2B or Q2R1. So Q2R1 is the old which is the chemical or biological chemical methods of method validation, development validation. But this is a bioanalytical method. There is a little difference between the analytical method validation and bioanalytical method validation. In analytical method validation, the normal parameters will be accuracy, precision, linearity, recovery range, all those things parameters are recovery, and then robustness, ruggedness. Apart from that, I want to encourage since the time remains another 10 15 minutes, so that I will touch on the little information about the thing. I will focus only the parameter which is not included in the analytical method validation. So bioanalytical method validation, it is a recent one. 2018, the US FDA has been given that drafted, and in 2022, it is also given the bioanalytical method validation with ICH M10 guidelines. ICH M10 guideline is a bioanalytical method validation. I'm going to focus on the part which is not covered by the analytical method validation. So the stage of drugs and metabolites in biology. So what is the difference between the analytical method validation? Analytical method validation is method validation for the drug which is present in the formulation or bulk. But bioanalytical method validation means the drug which is present in the biological fluids, for example, blood, CSF, urine, sweat, or lacrimal fluid. So between these two, there are a lot of differences are there because in the analytical method validation, the matrix will be very easy, and we know the matrix, we know the content of the matrix, everything. But in the bioanalytical method validation, content of matrix is complex because the blood sample will be there, urine sample will be there, because urine content will be there, blood content will be there. We don't know what is the content percent in the blood. We don't know what is the content percent in the urine. So all those things are there. So that's why the bioanalytical methodology is different from the analytical methodology. So the characteristic analytical procedure will be highlighted. The bioavailable studies for these things will be the bioanalytical validation is very important. And the sample will be first will be processing the sample, because in the normal analytical validation we are going to do the dilution. Here there is no dilution; it's called the sample processing. So sample will be processed into the different form. Actually, it will be freezing or it will be thawing. It will be freezing for a long time in the oxygen. Next, analytical technique. We have to choose analytical technique. Most of the analytical technique will be LCMS will be used for the analytical. It's used as an analytical technique for the biological method. Because liquid chromatography coupled with the mass spectroscopy will be most mostly adapted to the method for the analytical method, biological method. So method development and optimization. By using the LCMS, we have to develop a new method and optimize the method. Nowadays, many research scholars are focusing on the new method development by using the LCMS and method validation documentation. After that, method validation method will be possible, and then application of the method to the study of the pharmacokinetic parameters. So these are the different processes. And regulatory requirements of biotic methodology. These are the biotic methodology because the plasma is rarely involved. For example, the sample is a blood sample, plasma or serum. So the plasma or serum is involved in the HPLC and LCMS must need additional procedure to avoid the blood constituents. So that we have to go for the protein precipitation or solid phase extraction or liquid extraction to avoid the contents of the blood constituents. So these are the different types. Protein precipitation, we use ethanol or acetyl to avoid the protein precipitation. And then means protein precipitation method to avoid the blood constituents. Solid phase extraction. Solid phase extraction by using the common chromatography, and liquid extraction by using the polarity based mechanism by using different types of solvents. And then regulatory requirements of the sensitivity, reference standards, working standards, internal standards, calibration curve standards. These are all the already it is there in the analytical method validation. So this is common to analytical method validation. And the thing will be we have to go for the fixing of the concentration for linearity. So fixing of the concentration for linearity and range will be different. So we use called the LLOQ, means a lower limit of quantitation, upper limit of quantitation. So the lower limit of quantitation, first we must know that because here the system is involved. The system means our body system is involved. Since body system is involved, we have to find out the Cmax. Cmax, we should find out the Cmax of the compound. Means concentration maximum at particular time. So concentration maximum at particular time will be Cmax. So we must find out the Cmax. According to the Cmax, we have to fix the concentration for linearity. So once if you want to fix the Cmax, for example, if Cmax is 15 nanogram, then it may be 1 nanogram because the LQ is the 5% of Cmax. So you calculate this one, 5% of Cmax or less than, at least less than or less. So 5% of Cmax or less. So 5% of Cmax means, for example, if the Cmax is 15 nanogram, then the LLOQ should be 1 nanogram. Means 5% of 15 nanogram will be 2.5 nanogram. Less than 2.5 will be either it may be 1 nanogram or 2 nanogram. So that's why I given in the bracket. So in the upper limit will be upper limit of motivation will be 1.5 to 2 times of Cmax. So for example, if our Cmax is 15 nanogram, then our maximum concentration of the LLOQ means upper limit of motivation will be 100 nanogram. 2 times. Means we can have a 2 times 50 into 2 will be 100 nanogram. And will be Cmax will be 3 nanogram. Lower limit will be first standard and upper limit will be 8 standard. Total 8 concentration will be used in the calibration method, or it is called the linearity method. So Cmax with the 50 nanogram means lower limit of quantization will be first standard and upper limit of quantization will be 8 standard. Apart from this, quality control sample also will be included. So quality control sample will be lower limit of quantification with the quality control sample will be half of the LLVQ. So the LLVQ is here 1 nanogram. So since 1 nanogram, here the half of the LLVQ will be 0.5 nanogram. So we fix as a 50 nanogram. So the half of the LLVQ will be 0.5 nanogram. Then 3 times of the LLVQ will be 3 nanogram. So LLVQ is 1 nanogram. 3 times of LLVQ will be 3 nanogram. And 30 to 50% of the ULVQ will be 3 nanogram. Means ULVQ is 100 nanogram. Since ULVQ is 100 nanogram means 30 to 50% of the concentration will be 50 nanogram. And then LLVQ, ULVQ is 75% more than 85, more than 75% of ULVQ. ULVQ is 100 nanogram. Since ULVQ is 100 nanogram, then more than 75 we can fix as a 85 nanogram. So these three concentration also will be included in the linearity range. So previous 8 concentration standard 1 to 8 plus these 3, so 11 concentrations. So this calibration curve and quality control standards will be included. So the 8 standards 1 nanogram, 2 nanogram, 4 nanogram, 10 nanogram, 20 nanogram, 30, 70 and 100 nanograms are the 8 concentration of intervals between the for the calibration curve, and the 3 quality control standards also included 3, 50 and 85. 350, 85 is from this quality control standards. So this is a way of fixing the concentration for the linearity curve. So this is different from linearity of analytical method evaluation, because in the analytical method evaluation, according to the response of the instrument, we fix the concentration. We fix the concentration. அந்த பக்கம் போங்கய்யா. பாப்பா இங்க so at least 67% of quality control samples will be used, because when you see the limit of reduction, limit of concentration, plus or minus 15% and plus or minus 15% will be used for the quality control test for the different ranges. So bioanalytical method validation parameters. Actually, in this parameter, due to time confinement, I will cover the carryover because see the parameters: selectivity, specificity, carryover, precision and accuracy, robustness and ruggedness, matrix effect, recovery, dilution integrity, and reinjection. So reinjection and solution stability. So this actually this selectivity, specificity, precision, accuracy, robustness and ruggedness will be covered in the already it is covered in the analytical method validation, and recovery also covered in the analytical method validation, because very common parameters, these are very common parameters. But the peculiar thing will be carryover, matrix effect, then dilution integrity and reinjection, also solution stability. So these are all the things. I will cover these things. Actually, selectivity already but the thing will be only the selectivity acceptance criteria changes. In this selectivity acceptance criteria will be at RT, means at the retention time of analyte, less than 20% of the area of the LOQ, or less than 5% of the area of LOQ will be for the internal standard, and for the analyte will be less than 20% of the area of the LOQ. Specifically determines the analyte in presence of the UPOT matrix, and also the specificity also will be the same percentage. Actually, the procedure, everything is similar to that of analytical validation, but the only thing will be the limit will be the acceptance criteria will be different. So less than 20% for LLOQ and less than 5% for LLOQ of IS. For analyte will be less than 20%. For the intervention will be less than 5%. And appearance of the and actually the carryover is a peculiar thing in the analytical evaluation compared to analytical evaluation. So in this appearance of the analyte in signal in blank after the analysis of the sample with high concentration analyte. So the carryover will be better when you keep the sample in the any container, so that it will be having the same concentration of the container, so that it will be washing. So it should be appropriate washing. Avoid by using the appropriate washing, sample dilution and diluent. So the extend the duration of washing. So change the mobile phase and diluent. So the remedy will be thing. So the reason is actually it is an inappropriate washing. So there is a single word. Inappropriate washing means we are not washing properly and sample dilution will diluent. So if the dilution of the sample is less, more concentration is there, then it will be carried over to the next sample. It will carry over to the next sample. So that to avoid the carryover, so we extend the duration of washing, change of the mobile phase and also diluent. So you might be come across the same point in the area of fluorescence spectroscopy. In the fluorescence spectroscopy, the absorption of the sample into the cuvette will be avoided by using the complete washing. So that the higher concentration of the drug is absorbed onto the walls of the cuvette, it will be interfering with the next determination. It will be interfering with the next determination. To avoid that, we have to wash the cuvette with the relevant or with the mobile face or with the any other washing liquids or any distilled. And accuracy and precision will be common to all the things. And this is a measurement of precise but not accurate. So this is a diagrammatic representation of the accuracy and precision. And it will be easy to understand about the thing. So this is precise. What is precision? Precision means this is the target. Our center point is a target. So since we are not getting the center point, so it is not accurate. But it is precise. We are getting same value at the not accurate area. Because for example, our target is 100%. If you are getting the 99, sorry, if you are getting the 70, 75, 72, 73, or 69%, then it will be, it is called a not accurate. And this is a measurement of are accurate but not precise. But this case not be possible. But this case is possible. Second case, measurement of the neither accurate nor precise, because the results are distributed in the all the area. 10% of the 30%, 40%, 50%. If you are repeating the experiment, we are getting the different result. So this is thing, and this is accurate and precise. So this measurement is precise and accurate, both precise and accurate. So target is 100%, you are getting 99%, 99.5, 101.5%. And this is a best method is best. So when you get this result like this, the method is precise and accurate. We can conclude the method is precise and accurate. And Roberts is similar to the mathematical relation. Recovery also is similar to that, only the acceptance criteria is different, 10%. And then dilution degree and reinjection degree is another area. So dilution degree is to assess the ability of the method to quantify the analyte and yield accurate range of calibration curve. So that's because for each and every analyte, it will be diluted to a particular level. You cannot dilute the lesser range and also you cannot dilute in the higher range also. So that can be the range will be fixed to find out the dilution degree. So at what extent it can be possible to maintain the dilution property, then we can fix the dilution degree. So for each and every drug, there will be the dilution degree for 0.5 to 100 nanogram for working, because here it involves many other matrix also will be possible. So reinjection frequency will be, because to achieve the accuracy and precision upon the reinjection of the same dilution, so the reinjection must be the same result. So 3 batches in the 2 days. So different time, different days will be reinjection of the thing. We should get the reproducible result. And stability. So stability means the sample will be freezed and sample will be converted to the normal temperature, thawing, so freezing and thawing temperature. So chemical stability can be achieved by because sample will be estimated by using different types of environment. For example, freezing environment and a normal temperature environment. I have two temperature environment. Benchtop stability, how much time it will be stable in the during the experiment is doing. For example, once if you want to do the experiment, it may take half an hour, it may take 1 hour, it may take 2 hours also. So up to what time the sample will be stable? So that is called benchtop stability. So stock solution stability. So how long the stock solution can be maintained? Either it may be 6 hours or 12 hours. So even the stock solution is stable for 1 or 12 hours. We need not prepare again the stock solution, so that we have to find out the stock solution stability. Long term stability, even 2 days or 3 days it is stable. Then we need not work on the different types of solvent system, and we need not waste the solvent system for the preparation of the solution. so that we can maintain the solution for a long time. So processing of the sample stability. So processed sample stability, which process sample freezing and thawing stability. So processed sample drug sample drug will be available in blood or urine or any other biological fluid that will be converted to different form of the film. So that is called processed sample stability. So after the processing of the sample, whether it is stable or not, it will be find out. So this I conclude my talk. And nowadays the pharmacopea is also available online. You can go to the USP, www.usp.com. Nowadays many old versions are available. USP 3128, USP 28 EP, extra pharmacopoeia 65, and CP will be our Chinese pharmacopoeia. So Chinese pharmacopoeia will be in the form of Chinese language. We can find out. And other monographs also. That means USB, if you want to get the USB old monograph, you can get the data from the online pharmacopoeia. It is available. The online pharmacopoeia is additional information for the people who want to search the pharmacopoeia information. So, but latest pharmacopoeia is not available. Only the USB 31, 32, 29, all these things are available. So the website name will be www.usbp.com. But the website name is USBP, but BP is not available recently. Recently, BP is also not available. Only USB older versions are available. This is our additional information. With this, I conclude my talk, and I thank for the given opportunity. I thank for Mallarity College of Pharmacy for the given opportunity. I thank the principal. I thank my friend, student, Mrs. Divya Yada, for the given opportunity. Thank you.
On role of regulatory affairs and bioanalytical method.
I welcome any enquiries from the participants.
Thank you very much, Kannappan sir. It's a very useful lecture. Definitely, it's the present day for all the faculty. It's very much useful because regulatory affairs is the emerging topic nowadays. So it will be very much helpful for all the faculty because we have to train the trainers. That's most important. That's why in that concept we have started this one. You're all helping us like anything. Thank you very much. Thank you.
Thank you. Thank you.
Thank you, sir, your presentation. At 2 o'clock, we'll have a session with Dr. Tulibali Shyam sir on plant-derived products. The link will be shared before the 10 minutes of session. Thank you all and kindly join at the time. Now the session ends.